Thank you.
Don't believe any of the lies about the snowboard part. The snowboard was not pretty. So good morning, everybody, and welcome back to everybody in the room, but also everybody online. We're grateful that you're here on a Saturday morning. We're excited about this day, right? This is always a day where we get to talk about all the things we're doing. Remember I said that hope and optimism come from the things that we are doing, but also the things that we're working towards, and that's the theme for this morning. So obviously we're going to have a medical and a research panel, always one of the top highlights of VLC. We're going to dive in deeper into some of the really cutting-edge work that we're doing in research, and as well as focus on some of the latest clinical care developments so you can hear about the progress across the board, all right? So, I said this before, but I'll just get a little bit of an introduction, then we're going to bring everybody out, but certainly we're experiencing a shift in the CF and what it means to live with cystic fibrosis. And as people with CF live longer, the overall population, number of people with CF continues to increase. Again, one of those little pearl numbers, the 40,000 number you've heard of people in the US that live with cystic fibrosis now. That's a 25% increase over the last decade in the number of people with CF. This is thanks to the tremendous advances in research and in care. That's key. That's also led to more adults, right? We said 60% of all people with CF now are 18 years of age or older. That's continued to grow. And I loved yesterday talking about the milestones part, this whole idea of thinking differently about how all the milestones, or the normal milestones in many ways, are things that we want to have ahead for everyone with cystic fibrosis. I also shared yesterday that late-breaking, really late-breaking, as in yesterday, hot off the press, increase in the median predicted survival to 56 years of age. Exactly. Obviously excited about this. We expect this to continue to go up with all the progress we're making. But I did want to make one little comment about this, which is there is Andy Piper, who's here. I don't know if he's still here today, but who received the award last night. We were talking about our dreams for CF, and he mentioned something that I thought was really insightful. His dream was when we can actually have a meeting where we don't talk about life expectancy at all. So I thought that was really, really amazing. And this is all possible because of the decades of hard work of so many people in this room, some of the people we're going to hear from, the perseverance, the determination to continue to advance our shared mission. So today you're going to hear and learn more about the ways that we're accelerating that mission and really working to make sure that all people with CF can benefit from transformative therapies, particularly those with rare and nonsense mutations. So not only are we continuing to develop the next generation of modulator therapies, we talked about that, but expand these therapies to more people with cystic fibrosis, to younger groups with CF, but really aggressively and boldly pursuing some of the most innovative science and the technologies that are just cutting edge throughout the world of science and applying them to our path to a cure, right? This path to a cure, this commitment to make sure that everybody with CF has transformative therapies, no matter what type of CF, no matter what mutations they have. So we've been encouraged as we've gone in this, and there's been a real breadth of opportunities that really all support the path to the cure. And we've been really proactive about deepening our collaborations with leading biotech and venture capital firms to make sure that we apply the world's most promising genetic technologies to cystic fibrosis. Sure, go ahead. We want you to be successful in sickle cell and other diseases, but we just want to make sure that cutting-edge technology is also focused on cystic fibrosis. So our Chief Scientific Officer, who you'll hear from shortly, Dr. Steven Rowe, is going to highlight these efforts and building sort of this ecosystem and strategy, be aggressive to make sure we accelerate the path to a cure. He's also going to discuss the Foundation's therapeutic development program sort of overall, our overall strategy on that. Obviously, at the end of the day, curing CF is our ultimate goal, but we do remain focused every day on addressing the challenges that people with CF still face in wherever they are in their journey, right? There's so many different journeys. Part of it is that we celebrate the growing number of adults with CF. We also know that the healthcare needs keep changing and become more complex, truthfully. So you're going to hear as well about not only the path to a cure, but the way that we've been trying to tailor the healthcare experience to all those different journeys that people are going through in CF. And then you're also going to hear some of the trends that we see in care and get sort of a sneak peek into the most current patient registry data, right? We have one of the world's best patient registry data. About 90% of people with cystic fibrosis in the United States are included in our patient registry because of the generosity and willingness to participate. We thank everyone who's watching who's part of the registry. But that allows us to identify trends. It allows us to identify areas that we need to work on, and you're going to get a little sneak peek of that. And won't be formally published for several months yet, but you're going to get a little, as I said, a little sneak peek. And then we've set aside plenty of time for Q&A, and looking forward to that part, which is always one of the best. So I'm going to introduce now the three speakers we're going to have. They're going to come up and they're going to each talk a little bit, give you an update in their areas, and then we're going to sit and take your questions. So as you know, the first speaker that I've mentioned before is Dr. Steven Rowe, who joined the foundation this past year as our new Chief Scientific Officer and Executive Vice President. And Steve was by far our #1 choice for this. As a matter of fact, I did a little arm twisting, but I'm so grateful that he was receptive to arm twisting because just a great leader for us. And Steve is respected globally as a leader in CF research. Many of you know his work or have been taken care of by him in the past. He brings more than 2 decades of experience to this role, including serving as a principal investigator for the first triple combination therapy, Trikafta. So obviously had a big impact in so many lives in this room. Steve oversees our basic and clinical research, and he leads the foundation's research strategy and the venture philanthropy programs, including the Path to a Cure work. Second, we're going to hear from Dr. Kara Foshay, who's the Senior Director of Genetic Therapy Programs, and she oversees and directly is involved with the companies in the Path to a Cure portfolio. So prior to joining the foundation in 2020, Kara was a neuroscience research investigator at the Inova Health System in North Virginia. And there she led brain tumor research team, actually, but we're glad that she decided to change her focus because she's fantastic. I mean, a great leader for us. You're going to really appreciate listening to her speak. And then last, one of our long-term favorites, somebody who's been a dear friend to me for many years, and that's Dr. Bruce Marshall, who's had such an impact on the care of CF. I call Bruce a quiet dynamo because quiet, but behind the scenes, just having incredible impact in the daily lives of people with cystic fibrosis through supporting really this amazing care network, right? So Bruce is our Chief Medical Officer and Executive Vice President, and he directs really all our clinically related activities, including the Care Center Network, our quality improvement work, our clinical practice guidelines, as well as the patient registry. So, like I say, that dynamo, there's a lot of things going on in the background, and he's been part of the foundation for 21 years. Before that, he was tenured faculty somewhat nearby, actually, at the University of Utah School of Medicine, where he was the founding director of the adult CF program there. So after the presentations, we'll do those three. We're going to open the floor to your questions, so start thinking about those, putting those in there as it was described by Kate and Mac. And I'm going to actually ask our panelists to go ahead and come on out. Thank you. Thank you, Dagan. Take it away.
Okay, great. Whoops, I think you're going to need to put my slides on the prompt, but I'll walk through it here and give you an update. So, hi, I'm Steven Rowe. So glad to be here with you. This is actually my second time here at the VLC, and it's even more fun for me on this round. And it's clear that we know how to have fun and with this positive celebratory atmosphere. And it's not just because we know how to dance in ski gear late into the night, or the fact that I absolutely crushed Mike on the virtual ski board, but rather it's because of this spirit that we've been able to be grateful for as we hear the stories of people with CF that are living in this new era of medicine, where by far the majority of individuals are on highly effective CFTR modulator therapy. So I want to start by focusing on what's happening with those individuals and the kind of research that we need to focus on moving forward. So this is shown another way. Perhaps you've seen it before. Again, emphasizing that we're up to 94% of people that have highly effective CFTR modulator therapy. And we're going to focus heavily on that top bar shortly. But we know that that purple group, the 94% of individuals, will continue to grow. And it will grow because we're still doing individual therapeutic studies to try and expand the number of patients that are eligible for these modulators. We also did a really important study called the Simplified, and it was really one of the highlights of this year's North American CF Conference, where we brought people all around the world to hear advances in CF care and research. It was entirely funded by the Cystic Fibrosis Foundation, so thank you for the support that has allowed us to do this. And what we did in this study is actually randomize people to discontinue one of their chronic therapies, either hypertonic saline shown on the top or Dornase alpha, what you might know as pulmozyme on the bottom. And you don't have to know what the fancy words like non-inferiority margin mean, but see that those points on the curve land right on the zero line, meaning that people appropriately selected can discontinue some of the therapies that take 2 or 3 hours a day to do and have no difference in their lung function at the end of the day. Can you imagine, I certainly could not 5 years ago, that we were now entering an era where we can actually think about simplifying the regimen for people with CF. There's a lot more to do. Who's the right group of patients that can do this? When do we need to reinstitute them? What do we need to be aware of going forward? But this was really opening a new era as of this year. The CFTR modulator landscape is also very robust, and we continue to work heavily in this area, obviously because of the success. One of the studies that's currently ongoing and soon we'll have the results of is a new single therapy Trikafta version. This is meaning patients can take 1 pill a day. Obviously, that will be a major advantage for people with CF, as anybody that's raised a child or a teenager knows that if we can improve the ability to take and use these medicines long term, that will be good for individuals. We also have other partners moving forward with other CFTR modulators, and that includes our colleagues at AbbVie in moving their own triple combination therapy, obviously having more choice and a different range of effects will certainly strengthen the armamentarium for people with CF. And we also have another program called Sionna Therapeutics, which is really born out of about 10 years of science. It's a topic I spoke about with the former CSO, Bill Scatch, about 8 years ago at the plenary, where we imagined new drugs that could fold the F508del protein more correctly in the nucleotide binding domain 1 region. What that means is we now have a new modality working extremely well in preclinical models that have been very predictive of what we might see in the clinic. And we can imagine that working in other mutations that are currently uncovered, and also based on the results we see in the lab, even more effective than what we've seen with Trikafta. So we're quite excited to see that program moving forward in patients and normal volunteers. Well, I want to touch on a couple other areas that you may not have heard of as prominently. And one that's quite interesting is a phage therapy. You may not know what a phage is, so I put a picture or two. Those are not spaceships or lunar landers. Those are actually bacteria. You may not know that we have 10 times more bacterial cells in our bodies than we actually have human cells. And with those 10 times more cells, there is actually a war happening all the time. And that war is between viruses or phages that only infect bacterial cells. They don't infect us, they're attacking these bacteria. And the bacteria are responding and going forth. That's happening all the time in us, and it's also happening in the lungs of people with CF. And so what we're conceiving now is the idea that we can take these phages and design them to go after the bacteria that are infecting the CF lung. First, you harvest the bacteria that's shown in the first emblem from left to right from the patients. Obviously, that's very easy for us to do with a sputum sample. Then we test them against hundreds, perhaps even thousands of phages, and then develop a cocktail that can be re-administered to the lungs or perhaps one day to the blood of people with CF, and then let those phages do the work and clear out. And we've already seen, in fact, promising results. These are early phase studies, our first studies in people with CF from 2 different programs that look favorable, and certainly enough to us to think about moving forward. And again, to try and achieve one of my dreams for people with CF in the intermediate term. And that is that we can give a medicine, whether it's an antibiotic or a phage, to people who are on CFTR modulator therapy and allow them to have a bacteria-free life of their lungs. Well, this is just a taste. Obviously, we're doing a ton of other work. You can see there's 49 infection programs, currently 21 that are funded, 28 under evaluation. We also have anti-inflammatories and other complication research ongoing, all to help us prepare for this next era of therapy of patients on highly effective CFTR modulator. Well, now I want to pivot to something that obviously gets us up in the morning and keeps us up awake at night, and that is how help the people that currently do not have CFTR modulator therapy. And the analogy I like to use is that we are close to the peak of Everest. You may have read the book Into Thin Air. There's obviously also movies and other interesting books about this. And the point is that we are the envy of many other diseases. And one of the most remarkable stories in medicine, we're nearly at the top, but that last push, that last climb to the summit is actually going to be one of the most challenging. We're preparing for the summit day, and I'm going to tell you just a taste of what we're doing to prepare, and you'll hear about the details from Dr. Foshay. Well, first of all, is to point out that on that same therapeutic development portfolio, we have 82 different programs that are currently either funded or under evaluation to try and push to this final task. And we're also improving and augmenting the ecosystem of research, developing new partners and tools that are going to be remarkable in helping accelerate our partners to try and achieve this. And another aspect of this is we've had a huge success of really using our bullhorn. Come to CF, we'll provide funding, we'll de-risk your studies, we'll have a clinical trial network ready for you. And what we're headed towards next in the path to a cure space is to have a very targeted relationships. There's obviously remarkable things happening in genetic therapy. The list of approved genetic therapies that can cure diseases like certain forms of hemophilia, soon sickle cell disease, other eye diseases is truly remarkable. The lung is going to be a much more difficult target. So we have to go out and find those people willing to take these risks and try a harder target like CF. And we're bringing to the field, well, how are we going to do that? Well, we've developed our therapeutics development program to really focus on this mission-focused strategy and are augmenting our team, building on our strengths, but also in entirely new areas. And that includes efforts to go out and find these partners, find out with a diligence effort which ones are the very best, which ones will be in it to win it, and then develop our deal structures and investment strategies to try and accelerate their progress. And then a really important aspect that really I'm importantly charged with is to really be the partner for these relationships, the partner for these companies to bring them to a successful conclusion. So we're expanding our resource toolkit in this new genetic therapy. We've always had strong academic partners. The CFF lab is actually bigger than ever and pivoting heavily towards the path to a cure space to try and essentially work hand in hand with our partners, whether from academia or companies working in the genetic therapy space to move these forward. We also have a number of resource infrastructure investments to try and be able to help do the experiments that are needed to bring these people to the field. So here in my last slide, appropriately for Denver, we'll talk about how we're going to climb the mountain to the path to a cure. And what I want to prepare you for is that we're going to have a number of important intermediate steps. And that summit day is going to be challenging, but it's going to be stepwise. And I think we have a lot of hope that we'll see substantial progress in the way before we get to the top. And that includes mRNA therapy for the lung, which Dr. Forshay is going to discuss, and why we're quite excited about that. Also, gene therapy and gene transfer to the lung, again, inhaled, and we already have studies ongoing on this. We know we're going to learn from them and continue to get better as we design newer technologies to try and achieve this. We're thinking ahead further. We're using the sorts of gene editing strategies that have been useful to be able to do outside the body in certain hematologic conditions or bloodborne conditions, and get ready to be able to do that in the lung for perhaps one mutation or two mutations that have currently uncovered, but will really set the proof of concept for that we can ultimately do this for everyone and cure all mutations. So if you don't believe that we can do this, I want to share this slide that Casey White shared with me at our last board meeting, and that is in fact that Nick Talbot, a person with CF, has summited Everest himself and obviously brought attention to the CF cause. So I know that working with you and working with the team that we have and that we are building, that we can ultimately accomplish this task. So to hear more about the details on this, I'm going to pass it off to Dr. Foshay.
Thank you, Steve. I am honored to be here and greatly appreciate the opportunity to share in your weekend, which has been emotional and fun, and I am just really, really thankful for the opportunity. Do we have a clicker? Which one? Oh, this?
Yep.
Sorry.
That's right. There you go, right there. Green.
So as Steve highlighted, the Path to a Cure initiative supports many different types of drug development programs. And initially, these will focus on the 6% of people with CF who are not eligible for highly effective modulators. And by the nature of their mutations, they need a genetic approach to treat the underlying cause of their disease. And so it's our hope that our learnings and our experience in this population will build and eventually translate to a curative therapy for all. The Path to a Cure initiative includes some mutation-specific strategies, such as NMD inhibition and read-through strategies for people with nonsense mutations, and ASO treatments for people with splice variants, frame shifts, and exon deletions. The Path to a Cure also includes additional theratyping efforts and a search for new and improved modulators, as Steve talked about. But today, what I really would like to highlight are 3 genetic therapy approaches that could eventually benefit all people with CF. The first is to restore CFTR protein where none exists. And this is really talking about mRNA-targeted therapies. The second is to replace the defective CFTR gene with DNA-targeted therapies. And finally, to fix the defective CFTR gene inside a patient's cells with DNA or gene editing technologies. To put these therapies into context, I think it's helpful to review the biology. Inside the nucleus of a cell, we have DNA that encodes genes, including the CFTR gene. DNA is transcribed or copied into mRNA. mRNA is then transported out of the nucleus and translated into protein. The protein is folded and can function within the cell. In the case of CF, CFTR is a membrane-bound ion channel that transports chloride. So modulators work on the protein. They can help CFTR fold, get to the membrane, and open and close appropriately. But if a patient has a mutation where a protein isn't made, CFTR modulators can't work, and therefore we need genetic-based therapies. mRNA therapies involve the delivery of a new mutation-free copy of the CFTR mRNA into the cell. The cell can use its natural processes to translate this mRNA into a functional CFTR protein. With gene therapy, the premise is the same. A new mutation-free copy of the DNA is delivered into the nucleus of the cell, and the cell uses its normal processes to transcribe and translate this into a functional CFTR protein. Gene editing is a little bit different because we're not actually delivering a nucleic acid. We're delivering enzymes or proteins that can change the DNA code within our cells. Again, the correction would be made in the DNA and would allow for transcription and translation of a mutation-free CFTR. Now, initially, we're targeting the lungs with these gene therapy approaches, but the hope is that eventually they will be delivered as systemic therapies and can improve function in all organ systems affected by CF. So as these technologies are maturing, and a few are actually in clinical trials now, we have a better sense of, if approved, what these therapies would look like for many of you who could take them and benefit from them. So I'll walk through what these might look like. mRNA therapy would be a transient therapy for all people with CF, regardless of mutation. And the dosing would likely be daily, every few days, or possibly weekly. And that's because mRNA is inherently an unstable molecule. It can be taken up by any cell, but it will also degrade quickly. These will be inhaled therapies to benefit the lung at first. Gene therapy is also a transient therapy. It would be amenable for all people with CF, but would last a little bit longer than an mRNA therapy. And that's because when DNA is taken up into a cell, it will persist for the life of that cell. Now, since we haven't seen a successful gene replacement therapy in the lung, we don't know how long that effect will last. And that'll be one of the focuses of the upcoming trials. Gene editing is a little bit different again because the technology here isn't ready for the clinic in CF. So this is a future goal. But the goal of gene editing therapies would be to correct the CFTR gene in the stem cells of all the organ systems affected by CF. By targeting the stem cells, we can make an edit that will be passed to all new cells that arise over the lifetime of a person. In this way, a gene editing therapy could represent a permanent cure for CF. Yeah. So by definition, genetic therapies are different than small molecule therapies like modulators. They're bigger and clunkier, and one of the major challenges is getting them to the correct cells. They can't just diffuse into all cells. So whether coming into the lung as an inhaled therapy or through the blood as a systemic therapy, I think the most formidable challenge of gene therapy is reaching the correct native CFTR expressing cells. These therapies have to get to the correct organ and stay there. We don't want off-target effects. They have to reach the appropriate cells. In the case of basal cells, you see those are located at the base of the epithelium and are sort of hard to reach. And they have to be redosed as the cells turn over. In addition to getting to the right place, these therapies have to get through pretty inhospitable environments. So there's thick, sticky CF mucus that I don't have to tell you all about. But there's also immune cells that are out to take up gene therapies and destroy them before they get to their targets. And so I think the take-home message for me is that gene therapies aren't just nucleic acids, but they also need protection. They need a delivery vehicle, and that makes them a very different type of therapy. So delivery vehicles are one of the major focuses of the Path to a Cure portfolio. This is a major oversimplification where I showed two main classes of delivery vehicles: viral vectors and non-viral nanoparticles. These delivery vehicles vary in the types of cargo and the size of cargo they can carry, meaning DNA or mRNA or even proteins. They vary in the immune responses they can elicit from the cells in your body, as well as how effectively they can be taken up into cells and then release their cargo to the area of the cell in which it needs to act. They also differ with respect to the ability to manufacture these at large scale at an affordable cost. And so one of the major focuses of our portfolio is diversifying the number and types of delivery vehicle programs we're working with. So where are we? I think the most exciting thing about this slide is that there are a lot of new therapies starting clinical trials in the late part of this year and early next year, both in the US and abroad. That includes an ASO therapy for 3849 plus 10 KB mutation, 3 mRNA therapies, which are encapsulated in lipid nanoparticles. VX522 552 is up and running and enrolling patients, which is very exciting. We have gene therapy programs. We have 4D Molecular with their AAV CFTR inhaled program, which is enrolling and has enrolled several patients. And we have a few other viral vector gene therapy programs in the pipeline. So my hope is that next year or 2 years from now, Mike will invite me back and I can share some exciting updates from all of these trials. For now, I'll highlight a sneak peek at 4D's data, which they shared at this year's NACFC. Cohort 1 enrolled 3 patients with CF who were given a single dose of 4D-710 as an aerosol. And what's particularly exciting to me is that this was safe and well tolerated in all 3 patients. And CFTR delta R, which is the gene they delivered, was detected as mRNA in the cells of all 3 patients. So this is a representative example. Yeah, so those brown dots, that's gene therapy at work. And we look forward to seeing more data come out of this trial. Yesterday, Joe O'Donnell said something that kind of resonated with me, and I won't get the words exactly right, but he said something like, there's no good science for CF that we won't pursue. And so just because we have trials ongoing doesn't mean we're gonna stop investing in new technologies, because we know that they will continue to improve and get better. In the mRNA realm, we're investing in programs to make longer lasting mRNA, so patients could get fewer, lower doses, something that's potentially better tolerated. In gene delivery, we know that redosing is a major problem. So we're investing in programs to help with redosing and to explore systemic delivery of gene therapy. In the gene editing space, we know that CRISPR and the most standard technologies are great at editing one mutation at a time. But we want to apply this to all mutations. And so we're investing in programs that could insert a functional CFTR gene into the genome and essentially cure all mutations at once with one approach. And so I'll leave you with this. This is a rapidly evolving field, and it is complex. And as scientists and clinicians at the foundation, we want to make sure that we move forward with you and in a conversation with you. So one of my roles outside the gene therapy portfolio is on the gene therapy working group. And I have not necessarily led, but I have helped my colleagues to make glossaries and one-pagers and frequently asked questions that clinicians, coordinators, patients, families can use to have a discussion about these new and exciting therapies together. And with that, I'll say that cure and care can't be separated, and I'm excited to introduce Dr. Bruce Marshall to talk to you about our evolving care model.
Thanks, Kara. Nice job. Thank you, Kara. I can't really see any of you. Those lights are so bright, but good morning to everyone here in Denver and those of you online. I still get nervous speaking in front of a crowd this size, so I'm gonna break the ice with a confession. I'm a data nerd. In fact, there's a rumor that I sleep with the latest registry report under my pillow. That is not true. The registry report is on my bedside table. and I carry it wherever I go. So you've been forewarned. Brace yourselves for the graphs that will follow. First, just as Mike mentioned, some insights into the 2022 registry data, not fully baked yet, but some interesting data I think you'll enjoy seeing. Talk a little bit about what we're doing in selected areas, and then close with some comments about evolving the CF care model. Mike talked about median predicted survival. You may have seen this graph. It goes back all the way to the left to the 1990 in 5-year increments. You can see back then median predicted survival about 30 years of age. And you heard yesterday, if you go back further, you get back to single-digit median predicted survival. And then gradual increase over time. And then with the availability of Trikafta, that upward trend towards the end there, 53 years of age in that 5-year increment between 2017 and 21. And this is where we are in 2018 to 2022. This latest 5-year time band, 56.6. This, like I say, not fully baked. This may change by 0.1 or 0.2, but it's definitely going to be over 56. So, now, when I look at this graph, I'm excited about the upward trajectory, but I think what comes to my mind is the many contributions from so many people, going back all the way to Dorothy Anderson, who first described the disease in 1938. And it starts with the parents who would not accept the poor prognosis they heard about their children with CF. They started the CF Foundation in the mid-50s, determined to change the course of the disease. I also see the countless hours of airway clearance, inhaled therapies, other treatments done day to day in the homes of people with CF and their families. I see important contributions of donors, volunteers, care team members, clinical researchers, basic scientists, many others, and of course all of you in this room and online. The underlying storyline on this simple line graph is what has been referred to as the greatest story in medicine. That's what I think about with this graph. And oh, by the way, one final point, 56 years is not good enough. We still have work to do. So with increasing life expectancy, there will be more adults with CF. So if you could cue up the video, please. Incredible to think about that projection forward to 2040. With about 75% of the patient population over the age of 18. Okay, more data. This is a slide showing the percentage of adults, adolescents, and children treated with IV antibiotics for a pulmonary exacerbation. And that line stayed pretty flat for many years until the availability of Trikafta, and you see in 2020 that marked drop. It was sustained in 2021, and pleased to report that it's sustained in 2022. What are we doing in this area? I think some of you know we've had a strong group of investigators focused on exacerbations for a number of years, working on standardizing treatment. STOP2 trial really defined the optimal treatment duration. STOP 365 is now enrolling, and it's going to compare the outcomes of 1 versus 2 antibiotics. This is important. If it's a positive result, we may be able to drop out one of those antibiotics that can cause some toxicity, hearing loss, and on occasion, kidney damage. So important work in this area. This slide shows the number of lung transplants from 91 through to 21. And you can see over time that gradual increase in the number of transplants and then that marked drop with the availability of Trikafta in 2020, again in 21. And again, we see this progress sustained in 2022, about 56 transplants. It may wiggle by a few, but mid-50s. What are we doing in this area? You'll be relieved to hear that I'm not going to go through this slide in detail. But the point that I wanted to make is that we've built a robust program around advanced lung disease and lung transplantation that includes the development of guidelines. These are difficult to do, registry data, research, and making tremendous progress. You might say, well, why are we still interested in transplant? Well, we believe that lung transplantation will remain a viable, life-saving option for some people with CF. Some people with advanced lung disease benefiting from Trikafta, they may need transplant at a later stage of life, and they may have more comorbidities, other health situations that have to be navigated through a transplant. The other point to make is over 1,600 people in the US with CF are living with transplanted lungs. They have complex medical issues that need high quality, coordinated care. They also need therapies to prevent and or treat chronic rejection. One final point on this slide is March's colorectal cancer awareness. If you're 45 years of age or older, talk to your doctor about this. I bring it up on this slide because patients post-transplant are at higher risk of colon cancer. So we're promoting this to this community. If you're over 30 years of age, talk to your doctor about colonoscopy. Are you still with me with the graphs, with the slides? Okay. This next slide shows the number of pregnancies born to women aged 14 to 45 years of age. And you can see, again, with the availability of Trikafta, big bump up, and again, sustained in 2022. The opportunity for people with CF to have a family is certainly a positive, but we know that some of these pregnancies were not planned. We know that some care teams have little experience in this area, so we have work to do in this area. Fortunately, we have a very strong working group on sexual health, reproduction, and gender research, co-led by Drs. Jane Taylor-Crusar and Kazmersky. They've helped us develop this clinical considerations document that we've distributed to the Care Center Network, and they're also very active in research. The Mayflower study on the effects of CFTR modulators on women with CF during and after pregnancy, and the HOPE study on the impact of parenthood on the health of women with CF. This next slide is about mental health. Mental health remains a concern. We have data on anxiety and depression in adults and adolescents. Part of this increase that you're seeing, we believe, is due to the implementation of systematic screening. When you look for things, you find them. But we see in adolescents here on the right, particularly in anxiety, an increase in anxiety. And we know that the pandemic has been very difficult for adolescents and young adults. So we're committed to furthering our work in mental health and offering mental health care to everyone with cystic fibrosis. We've done a lot in this space. We committed to this a number of years ago, and I won't go through this in detail, other than a shout out to the amazing contributions of the Mental Health Advisory Committee. We've worked closely with them, and they've just done amazing work. Also of note, on mental health, it's been an international collaboration. We've shared resources, we've shared learning, and that international collaboration continues to this day. So much more work in this area, but we're strongly committed to it. And this, we love this sort of mantra from the World Health Organization: there is no health without mental health. This next slide is about nutritional status for adults with CF. And if you go back to when I started in CF care and research, about 30% of the population was underweight. And now you can see these categories: purple, underweight; blue, normal; yellow, overweight; and red, obese. These are defined by body mass index. You can see the underweight percentage has come down. And then the trend that we've noticed, again, accelerated a bit with CFTR modulators, is more in the overweight and obese categories. Not much change in 2022, but we're closely following these trends. Why? Because obesity is associated with hypertension, type 2 diabetes, hyperlipidemia, heart disease, and other complications. We want to prevent that. We don't want the CF population to look like the general US population, where obesity is about 40% of the US population. What are we doing in this area? We pulled a multidisciplinary group together to develop a position paper. There really wasn't enough data to publish a guidelines. And they pointed out to us, it's not as simple as eat a healthy diet and exercise. Obesity is really a complex issue, often associated with stigma, issues with body image, eating disorders, etc. In this document, they've published an algorithm for weight management, and they've recommended regular blood pressure checks and lipid panels. And these are data elements that we'll be adding to the registry to track. Okay, no more graphs. I'm done with the graphs. One other study, strong CF study, is in development, and this is looking at nutritional outcomes in adults and looking for metrics in addition to body mass index, which is a crude metric. So I'll close with just a few comments about the care model. And remember, the care model was developed in the mid to late 60s, proactive care, multidisciplinary care, but it has not been static. A number of innovations have been introduced over the years, and I won't go through this list. These are some of the ones that I thought, well, maybe they've been the most impactful. This image shows that it takes a village to take care of people with CF. We have required team members and recommended team members. And this team, we're going to need to reconfigure it as the needs of the patient population evolve. And we've already started on this work. We've started to recruit gastroenterologists, endocrinologists, and we have on our roadmap others where we're going to need to pull in that expertise, including primary care physicians. And this is my final slide, talking about the evolving care model for an aging and more diverse population. And I'll just present some of the work that's underway. Real-world research studies underway, focused primarily on virtual care. We're in the midst of planning for a comprehensive survey of care center staff, people with CF and their families. Getting this information from these key stakeholders is going to be very important. We're forming a task force on care center staffing and clinic visits, And I'm sure there'll be other issues that we'll tackle with that task force. And then we've just started in-depth planning for the next generation of the patient registry, and we've made a really important hire to help us lead this work. So with that, thank you for the attention, and I think, Mike, I turn it back over to you. Thanks.
Well, I think as you listen, you can get a feel for the amount of work that's going on in between meetings, right? And again, really encouraging. So lots of questions. One thing I'm going to give you guys credit for is that as I was watching some of the questions pop up, there were some that were asked, and then 2 slides later, you actually addressed them. So that was well done. So let's start off with just diving in. Please, for people online, people in the room, please go ahead and enter your questions. Things that we can't get together, we're not going to be able to cover everything, but anything we don't, we'll think about other ways of being able to give those answers out in other ways. Steve, I'll start with you. We'll work from one side to the other. And one of the questions came from our friend Linda Bowman, I'm sure is online. It was about theratyping. So you talked about expanding the number of people who might be eligible for modulators or be able to benefit from modulators. And she asked specifically about sort of the role of theratyping. Should we have more centers doing this? And I know we've had some recent things in the lab to talk about. So
Sure,
yeah.
sure. Glad to talk about that. So for those of you that may not be familiar with this term of theratyping, it's the idea of that we could take somebody with an unusual CFTR genotype and then test to see, does it work with therapy A, B, or C, perhaps some of the approved therapies or therapies that are in development, and give us some a way to help them obtain access. So there's a bunch of areas that we're working on in this regard. So one of the most important ones is to recognize that it was really a hand-in-hand relationship with the team at Vertex that allowed us and them to go to the FDA to then add medications, add mutations to the label, even though clinical studies had not been done in those individual populations. And that was really a remarkable event that the data that we had generated to generate the connection between what was happening in the dish and what could happen in people has really aligned for. So what are we going to do next? Well, one is that we've completed a major effort, a multi-year effort to look at the top next 100 or so mutations for people with CF. And because if you rank order these, there's a greater number of people with the first one than there is with the second one. It's even much a greater proportion than that 100 mutations that you would think of that would be affected by these results. So we developed a 3-lab effort where 3 independent groups all tested this same group of 100 mutations. They then combined the results, and then that was all both either conducted or analyzed by the CFF laboratory, and is now about to go online that will provide a guidance in a few ways. Number one is individual providers can then look at that data, make good decisions for the person with CF that they're treating, and generate a data-driven request to then use Trikafta or other CFTR modulator regimens for those individuals. The second is, of course, we're communicating, in this case with Vertex, where they can then look at what data has been generated and then accelerate their efforts that they've used in coordination with the FDA to get those officially on the label. And of course, that would make it easiest to access those medications. That same relationship we're fostering with the other CFTR modulator companies that are in development to accelerate that. And that's one reason we want to make these data publicly available in the very near future. Finally, I think we know that as you go beyond that list of 100, that there will be mutations that are so rare that they didn't come to our attention, but of course very much on the minds of the individuals that have them and the physicians that are treating them. And so what we can expect going forward there is to try and develop systematic algorithms to then take those individual cells and then look at them in the dish to see if they respond, and then couple that with an early clinical study to see if they respond by, say, a sweat chloride measure or a lung function measure. And then as we develop a more and more robust data set, then we can imagine that being a path forward for patients with rare mutations. I wrote an editorial in the journal Nature that talked about this and how we might use sweat chloride going forward. And J.P. Clancy's leading efforts in our multiple academic labs that are really fostering this approach so we can ultimately get there. So lots of things happening in this regard. Some are on the short term, some are on the intermediate term, but we will definitely not stop until we understand all people's mutations and whether these incredible drugs can be used to help them.
Great, thank you, thank you. So Linda, I think you asked specifically about the more centers doing this, but soon there's actually going to be a list that's published by our lab of this large number of mutations. You want to look at that. If not, it's better to actually use some of those reference centers and do some of the places, because this is a tough test. You can't just sort of do this in a whole bunch of centers. We have those specific centers. So wait for that list to come out. You're going to take a look at that, talk with your doctor about that. But then if not, then think about getting connected with the Cincinnati group or the Alabama group to do some of that theratyping. So great. Kara, I'm going to move to next. I'm going to ask you two smaller questions at the same time. Is that right? So one of these was, there's 3 RNA programs, and we're supporting all 3 of those. So why? I think one of those is why 3? The other was, why not just give the cells protein? Could we actually just give the CFTR protein into cells rather than do the RNA and the DNA?
Yeah, so great questions. So we are supporting multiple mRNA programs, and that's because gene therapies are complex. As I mentioned, they all involve a delivery vehicle as well as that nucleotide-based cargo. And so there are differences between these 3 programs. And they may have potential differences in how effectively they get taken up into cells, how effectively that mRNA is made into protein, and how effective they are at improving clinical outcomes. And so I think we are not ready to limit ourselves, and we want to let all the companies that move forward with solid preclinical data, solid evidence of the potential to work, to move forward into clinical trials. And the second question is, could we deliver protein? The answer is yes. Protein can be delivered. In fact, in gene editing therapies, one strategy is to deliver editing proteins as protein. Yeah. But I think that it is a more straightforward approach to deliver mRNA because multiple proteins can be made from a single mRNA. If we deliver protein, the amount of protein we would have to get into the cell and get positioned in the right place in the cell would be very challenging. So I think our chance of reaching that effective dose of enough CFTR protein copies to be effective is better with mRNA therapy.
Yeah, that's great. And remember, it's not just sort of dumping it in there and mixing it around like tea, right? It's got to be in the exact spot. And it's got to act as a channel, so it's not quite as easy as just sort of adding it in like Splenda or something. Bruce, very interesting question. So the simplified data, which we were actually sort of, obviously excited about, that was the data that says, hey, if you're on Trikafta, you have modest lung disease, you might be able to remove some of your medicines. Very perceptive question that says, that's 6 weeks of data. Are there additional studies or anything that are going on which might look at some of the longer term outlook on what it means if you pull back some of those medications?
Yeah, Mike, there are 2 studies. There's one in the UK, I think it's called CF Storm. It's a registry-based study where they're tracking who goes on modulators and what's discontinued by the care team and the individuals with CF. And then we have a study called HERO-2. It's an observational study where the subjects that enroll, people with CF that enroll, have an app that they can tap in the treatments that they're taking that day, that week, and then that data will be connected to our registry data. And we know that people are juggling their regimen around. And we've seen pretty good follow-through on the people enrolled in that study we'll be able to see their changes in regimen and look at some of the outcomes in the registry data. And it's a big study. I mean, this is, I think, over 800 subjects enrolled. So it's not going to be as clean as Simplify, which was rigorously designed and very close follow-up. This is real-world data, but we're hoping that we can learn something from that study.
Great. Important data to have. Thank you. Steve, I'm going to actually ask you one that I think everybody in the room probably has thought, but maybe didn't ask, which is sometimes we talk about the last 10%. Sometimes we look at the slide, it says 94% are covered. Talk to us a little bit about that 6%, 10%. What should we
Yeah,
say?
it's imperfect, and that's why you see it vary, and we've all struggled with it. So we sometimes we think about it in terms of people. Sometimes we think about it in percentage of mutations that are affected. Sometimes we try and cross-coordinate that. Sometimes we're thinking about the US data where we have the most rigorous assessment of how many mutations are out there. Sometimes we're thinking about the world where obviously the proportion of mutations are not. So that's why you've seen some of the variants as we struggled to head that. The second thing is that we're also cautious, like for example, the age of approval for even the various CFTR modulators that are already available are different depending on which one, and they're changing all the time as we do better and better to start to test these and prove that they're safe and efficacious for our youngest babies. So all of those reasons start to layer into what that proportion shows. What we're settling on now is to really think about that in terms of the genotype that's affected, and that's what you saw on our slide, and what we've used at the North American Plenary in the last year, and that's where we're headed to. But we're also cognizant that there are a couple other factors that are really, really important. One is that beyond what I've mentioned, and that is that there are people that are on the label but are not able to tolerate the medications, and we've not forgotten about them. They're very much on our mind. That's a painful position to be, and so. That's why when in practical terms, although when we look just at the genes affected, we might be at 94%. If we look at the practical things of who can actually take them, et cetera, that number is bigger. And so we need to work harder and do better to bring that number up.
That's great. And that key distinction there is between sort of the gene part and then sort of the practical, especially as everybody who takes care of patients has some patients who can't take it because of some side effects. Right, so that's why we oftentimes say 10% of people still not able to benefit from modulators. That's a rough estimate. That's not based on just the genotype. That's on real life in some ways. And I'm sure we have loved ones in this room who can't take it because of liver function abnormalities or other side effects. So that's why that number is wide there. Kara, I'm going to ask you one, which I think is a really good one, that it gets back to this sort of difference between systemic and lung treatment. And the specific question was, I've heard the true cure being described as walk into the doctor's office one day and, with CF and walk out. And is that really what that would look like? I guess what does a one-time cure look like? I think really they're asking about if you had systemic therapy with gene editing, what could you actually, what's our top point, or what do we have as a possibility there?
Yeah, so I mean, that's a good question, first of all. We don't know. So I think we love the idea of delivering an editing strategy that could edit enough cells, so editing CFTR in vivo in your cells, and edit enough cells that it could be a one-and-done cure. In my opinion, realistically, it'll probably take a couple doses. I don't think it's a one visit. It's okay. Well, maybe a handful.
We're willing to make 2 or 3 visits.
A handful of visits, right? The other thing is that these editing strategies will likely target stem cells. So as you live, your organs constantly turn over, and these stem cells replace and renew the cells of the organs. So if we edit only stem cells, there will be a lag as those cells make new cells and repopulate the organs. We need enough cells expressing the edited, corrected version of CFTR to see an effect. So I don't think it would be you're cured that day, everything is better, but I think it would be a gradual improvement over time that would eventually hit a plateau where you have enough edited cells in your body to maintain that effect over time.
And really, that would be a true cure.
Yeah.
Obviously, the whole other area we always talk about is restoring damaged lungs. That's another area. But this would be the idea of actually getting it so that most of the cells in your body didn't have CF, or at least in the tissues where it's really needed. So I'm going to actually double back on one thing, because there was a comment here I thought was really interesting. That 10% or that 6% number, this really is based on US data. All right, so we base this on registry, we base it on and as we start to look more worldwide, that 10% number is probably much bigger. And that's particularly because in that last group, we know that people of color are more likely to not be able to benefit from modulators, and there's more rare mutations and nonsense mutations spread around the world. So if you look worldwide, that 10% number is an underestimate. I think it's a key distinction, and thank you for the comment in there. Bruce, one of the questions that came up was about growing older with cystic fibrosis. You mentioned a little about colon cancer, but are there other things that we're doing or sort of paying attention to, or sort of thinking ahead about hey, this is a group who's maybe facing some of the other adult challenges that we used to not have to worry about as much?
Yeah, absolutely, Mike. When we think about evolving the care model, that's one of the big factors, the aging of the population. And we recognize as people grow older, other health issues arise. And we anticipate there are going to be issues that are completely separate from the cystic fibrosis, like hypertension or hyperlipidemia or heart disease. So we need to anticipate that. And that's the point about primary care. Most of the leaders at our care centers, physician leaders, are pulmonologists. And like myself, Steve, and you, Mike, we've treated hypertension, but it's been a while. We've treated hyperlipidemia, but it's been a while. So we're thinking about, do we have an AP, like a nurse practitioner or physician's assistant as part of the team that could help with primary care, or is it a referral out to a primary care physician? And then you've got to work on the coordination to make sure that the left hand knows what the right hand's doing. So definitely thinking about that. That's one of the big factors with evolving the care model.
Especially if we're going to get those geriatric CF clinics that someone suggested the other day. That's right. There's a second part of that question that actually it's more of a comment. It says, shout out to Dr. Rowe, who treated me at UAB when my CF was diagnosed at age 57. So there you go, Steve. Steve, one of the things you talked about was sort of being innovative in some of our approaches and our collaborations. And can you talk some about maybe some of the collaborations we have? Flagship Pioneering comes to mind as one where we sort of say, hey, we're going to try to do things a little bit differently. Yep. And sort of the reasons why for that, and not perfect, but there's some real exciting opportunities.
Yeah. Well, you heard a little bit from Dr. Forshay about how we're going to have to put different pieces of the puzzle together. First, we're going to have the right genetic editing technology to target and be able to replace and swap out the gene. We're going to have the right delivery technology that brings that protein machinery and the genetic machinery to the cells. And we're going to have them be able to work together. We may have to have other technologies, such as the ways to clean out and access a lung that's infected or has a mucus, to make those work better. So we're going to have to carve out the sorts of partnerships that are needed to cross all the dots. That doesn't mean we can't pursue each individually in the laboratory or in other experimental models to be able to accelerate that quickly. But ultimately, we're going to need to be able to be nimble at choosing and helping form partnerships. And the Pioneering Medicine flagship is one example, or perhaps our first example, where we're trying to do exactly that. That involves 3 different companies under a single umbrella called Pioneering Medicine. One is a genetic editing technology. One is an RNA company that's making and delivering a novel RNA that wasn't on Kara's slide because it's too early to put it there, but we're trying to accelerate that. And then one is a company that is developing a novel particle delivery technology. So we're helping them work together. We're doing experiments with each of them. We're doing experiments where we're combining the two modalities. So this is our first real push for this, but we know that we're going to need to do more and to help the sort of collaborations occur that can push us over the top.
Perfect. Thank you. Kara, I think, obviously we are excited about all the opportunities. We want to be talking about all those things that are potential, but we also want to be realistic about the challenges. And even some of the things that we say, hey, you have to be extra careful about. So one of the questions is about what are some of the potential side effects, for instance, of inhaled mRNA. But I'm going to expand it out. So what are some of the things across all the genetic technologies that we just have to say, we have to be real careful about this before we go charging in, or at least make sure we're monitoring along the way.
Yeah, absolutely. Well, I think most of us in this room have had an mRNA therapy. We've had our COVID vaccines, and we know that they can generate an immune response. And in the case of a vaccine, that's what we want. We want our body to make an immune response. But when we inhale a genetic therapy, we don't want to have an immune response. And I think one challenge is understanding what's going to happen when we breathe in those viral vector particles or those lipid nanoparticles. And each of these mRNA programs moving forward has a different lipid nanoparticle. They're not the same. And so the bodies may not respond the same to all of them. So I think understanding that immune response and what it's going to look like is a major challenge. And figuring out the frequency of dosing. So how often can we inhale those particles? How often is too often? We don't want them to accumulate in the lung. We want them to be cleared before we get a new dose. So I think that delivery piece really stands out as the challenge that pans or across all of the modalities, is how do we get enough of that genetic therapy in a safe and effective manner? So that's why we're focusing so much on delivery. That's really the major challenge.
And the lungs have special challenges,
That's right?
Because the lungs are, if our lungs easily accepted new DNA and RNA, we'd be sick all the time because we're breathing it in. It's very different if you're targeting the eye or the liver or sort of these protected organs. It's why actually CF is harder. It's why we need to be more aggressive. Companies actually want to go somewhere else that's easier. But what we say is, hey, we're going to help you. Come do the hard thing. We're going to partner with you. We're going to support. We're going to make it possible. So, did you want to add something?
Yeah, I was just going to say that we have real tangible experience now about this real challenge that we're trying to face. I was the principal investigator of the Translate Bio experience, which was yet a fourth mRNA, and it helps answer another question that Dr. Forshay says you did nicely with, is that we were quite excited about that, but ultimately it didn't work. There were a couple reasons, but one is that some of the people had immune reactions, and that didn't allow the dose to go up as high as we initially conceived. And so that's why we're thinking about alternate particle deliveries that might avoid that. That's why we're thinking about even immune system effects that we could then tamp down immunity for a temporary period to get that effectively delivered. It's the sort of learnings that we're going to have on the way, and it's emblematic of how this we've got a little tough road. It's going to be a hard summit, but not one we're afraid of.
Yeah. And I might actually say let's spend a little bit more time talking about the RNA, because I think the next 2 years are going to tell us a lot about RNA, and this is going to be really a key decision point about some of the therapy parts, because if the RNA programs look promising, obviously that's going to quickly get us to we have some therapy for everybody, no matter what mutations you have. And there's probably 3 categories of where those may play out. One would be a home run, where people have dramatic response, they tolerate it. That's what we're all hoping for, right? The other would be something in between, where we say, hey, we can actually get some expression, we see some signal there. It's not as much of the clinical response as we want, but we're sort of on that path. That actually is a win as well. It's still a big win, because that would allow us to say, okay, what do we need to do with delivery? What do we need to do with dosing? What do we need to do with some of the structure of the cargo to be able to make? The other one would be we say, oh, we continue to see problems. So people have immune responses, or they're not able to really see the expression. Obviously, we don't want that, but even if that's the case, we're going to do the same thing we did with Translate Bio, where you go back and say, okay, why? Why didn't that work, right? And so what is it that's causing the immune response? What is it that's allowing the cell to break that down? So that's going to be the big thing we'll probably be hearing a lot about in these next 2 or 3 years, which is going to provide a lot of directionality, because that's by far the one that has the most proximal, the closest opportunity in the genetic technologies to make a difference for everybody. Let's see, Bruce, I can't remember exactly where we are. I'm coming back to you for a second. How about that? One of the things that people asked about is, again, I like this because it's actually sort of the real life. Obviously, some people have had dramatic response to Trikafta, but even for people that have response to Trikafta but already had a lot of serious lung disease, right? People who maybe were waiting lung transplant, took Trikafta, and they were able to come off the list, which we've heard that story, they still have challenges. And what are we doing really in the, what I would say, advanced lung disease category, and also lung transplant, right? Because some of those people are going to progress onto lung transplant. Talk about the work we're doing there.
Yeah, it's a great question, Mike, and I glossed over that, but Al Farrow, who's here, and Aaron Tallarico, Abby Faxton, kind of lead this team focused on advanced lung disease and lung transplantation and palliative care, because not all people want to pursue transplantation. And there's a large body of work going on there, and recognizing folks with advanced lung disease, there are multiple health issues. Often they can't get all their care within the care center. So there's the coordination piece that's really important. And then with transplant, we worry that some of those folks that benefit from Trikafta, transplant may still be in the future for some of them at a point, where they've accrued some other health issues. So we're trying to anticipate that. What sort of care model do we need to kind of mold around them? So a lot of work going on there. I didn't really get into the research, but we've got a lung transplant consortium comprised of 15 of the best transplant programs in the country and in Canada. And they're just chipping away, developing the biorepositories to get the samples that are going to be needed and the data that's needed to really kind of map out the path forward for this sick group of patients.
Yeah, and I was just going to add on that there's also really formative basic research that's happening in this area that your support helps us support our academic labs to do that. Dr. John Sheridan, Patrick Thibodeau, who are in the audience, help run our stem cell therapy consortium. And one of that is just to understand the biology of repair, but the other is to start to queue up the potential learnings that would be needed to help turn cells that we already have into lung repair cells. And it's a little bit Star Trek because we have a long road to go, but so was CFTR modulators when we started investigating how the CFTR protein is processed and makes it through the cell. So that's really important basic work and that our team is pursuing to try and open those doors.
Yeah, great. And that answers that common question about could we actually help restore damaged lungs? So we have 5 minutes left. I'm going to take one question myself, just because I had been talking to somebody about this recently, which was asking about, there's been some really interesting articles about the effects of Trikafta during pregnancy, particularly so Trikafta is not approved for pregnancy right now, but there's certain times when you work with your doctor and say, guess what, even though we don't know everything, it makes sense to maintain the mother's health. And then the amazing part is, in some of those settings where the baby basically is diagnosed in utero with CF, amazing stories right now of those babies, mothers of a CF, and there's 2 last year, who are pregnant with a child with CF, that taking Trikafta actually prevents the baby in utero from developing CF. I mean, it's just incredible that this is really, really cutting edge. So when I say prevents CF, when the baby's born, they basically doesn't look like they have CF. Their pancreas is working. This is an area that we're just going to be learning a ton more about, but it's a tricky one because there's safety issues. And so I think the specific question is, what are we doing in this area? We actually do have a group, it's called Mayflower's group, which was looking at pregnancies to try to understand the effects both. We know that there was the Trikafta baby boom, where we went from 200 pregnancies to over 600 pregnancies in women with CF, right when Trikafta was approved. We have a lot to learn in this area, but we have a group that's focused on it, but it's not quite as easy as doing a clinical trial, particularly because safety always comes first. So what we're trying to do is to learn from real life experiences, but those type of stories that you hear, where you say, hey, a child is born, really can't tell they have CF because their mother's taking Trikafta. As long as they're breastfeeding and their mother's taking Trikafta, they're fine. As soon as they no longer getting Trikafta because obviously they're not breastfeeding, they're not in utero anymore, and they're too young to get Trikafta, they start to develop some of the signs. These are all stories about how important it is for us to continue to treat earlier and earlier and the potential benefit of that down the road. In the last 3 minutes or 4 minutes here, talk to me about something that you're excited about. So, Kara, we're definitely going to invite you back. So, but talk about things as you know, when maybe next year when we're back talking about these things, or 2 years from now, that you're excited about, because this is the things that we're going to be working on over this next year or two. And we'll start with Kara. We'll go to Bruce, and then come back to Steve.
Sure. So I'm obviously, like all of you, really excited about the mRNA therapies that we have coming. I think, you know, Mike, you alluded to this. These seem like the closest shot at a step towards successful genetic therapy. So I'm really excited to see how the Vertex trial pans out, the 40 molecular trial. I think we're going to have actual data to look at that can help us evolve our model. Right now, it's a lot of speculation. We think this preclinical study will predict success. We think this will lead to success, but we don't know. And once we have that data in our hands, we can use it to make a more educated next step. So I'm really excited to see how that moves forward. Whether it's positive or negative, we can learn and make a better step in the future. And I'm also really excited on the editing front. So I came here fresh from Precision Genome Editing Conference, where I talked to Nobel Prize winner Jennifer Doudna, and I went up and I said, hey, I would like to introduce myself. I'm from the CF Foundation. And she said, we love what you guys are doing.
Really? Yeah.
So
this is the Nobel Prize winner for editing, by the way.
Yes. Yeah. And so I'm excited about the partnerships that we are forming in the editing space with the leaders in gene editing, who are really going to make sure we are ready to move this forward into the clinic.
Yeah, I love that.
Yeah.
Bruce.
Mike, I confess I'm a data nerd, so I'm really excited about a lot of things, but the Next Generation Registry, I think, is a great opportunity for the foundation and the CF community. We're on a legacy platform now. We know we need to move, and there's a lot of opportunities to upgrade, so I'm excited about
So the
that.
Next Generation Patient Registry, which we're working on. We actually just hired a new
Head of data science.
he really is a data nerd. If he's watching, he's fantastic. But from MIT to help lead the next generation, right? And obviously a great team as well with Liz Cromwell and Alex. So they're working on the next generation of patient registry, which is going to not only give us more information, but I think it's going to help to guide care, right? At the point of care. So great. Steve, how about you, our Chief Scientific Officer?
All right, well, I just want to point out that I had to go last.
I know it's hardest.
And she took two. Just kidding around. There's a lot to be excited about. So one is what we're going to do next in the delivery technology space. And we really are, I already mentioned the Star Trek analogy, but I'm going to hit it again, is that there is some unbelievable science that's happening in how to target cells and how to package molecules, and it's going to be the new frontier. I really feel like when we, as I mentioned last year at this time, if there's absolutely no reason that CF cannot be the first one to effectively treat a genetic disease of the lung in a person.
Yeah,
And-
absolutely.
And so I'm really excited to see what these technologies can deliver, and the fact that we are doing outreach to find these nation ideas, couple them with academic investigators to bring these forward. It's going to be really fun to see what happens, and I'm sure we're going to get there, but it's going to be hard.
There we go. And I'm going to actually go last, which is, I usually talk about the science, but I think one of the things I'm most excited about is the opportunity to partner with you all in this. I mean, I think as we came out of COVID, we didn't know sort of how the work with the community was going to go, right? Are we still going to be together? We're going to be arm in arm, but I'll tell you what, after this meeting, I have zero doubts. We're in this together. We're going to make this happen. So that's the part that excites me. So guys, thank you. Absolutely. Wonderful. I think we can probably not